Hello, everyone, and welcome to today's webinar titled Gain Clarity in Complexity, Transforming Neurodegeneration Research with Biomarker Precision. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Olink Proteomics, part of Thermo Fisher Scientific. To learn more, please visit olink.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speakers for today's webinar. Evan Mills, Vice President, Proteomic Sciences, Thermo Fisher Scientific. Annalee Lind, Product Manager, Neuroscience, Proteomic Sciences, Thermo Fisher Scientific. And Jenny Rubin, Research and Development Scientist, Proteomic Sciences, Thermo Fisher Scientific. Evan, I will go ahead and hand it over to you first. You may now begin your presentation.
Hey, everybody. My name is Evan Mills. I'm the Global VP of Commercial here at Olink Proteomics, part of Thermo Fisher Scientific. Really excited that you decided to spend some of your time with us today. I personally have a background in next-gen proteomics for over a decade, and I used to do some neuroscience research. So I think this product is super, super important, both for us as a company, but more importantly, to the field of neurodegeneration research. So here to talk a bit about the launch of the Olink Target 48 Neurodegeneration Panel. Like all of our products, this is powered by the PEA technology, which uniquely uses a 3-factor authentication system to ensure specific, highly reproducible results. And this panel is the first time that we've actually launched a phosphoassay, p-tau217, given its importance and an emerging role as a neurodegeneration biomarker. And I think the reason this product matters to the field is that everyone's had to make a lot of trade-offs and had to deal with, suboptimal solutions. And our attempt here, after talking to a lot of you in the field and KOLs, was to come up with a unique offering that offers solutions that aren't available today. Specifically, the ability to measure 41 neurodegeneration biomarkers with a super small amount of plasma, CSF, or whatever matrix you want to use. 1 microliter is all we require. And we're delivering this very well curated list from, again, very small volumes in absolute quantification, right? So gone are the days where you have to connect multiple smaller panels in absolute quant to look at the range of biological functions that are relevant for neurodegeneration. With this product, you can do it all using super small volumes, get a tremendously broad view of neurodegeneration biomarkers in absolute quantification. So, depending on what your goals are, whether it's measuring, samples from cohorts, your own clinical cohorts, some smaller preclinical samples, drug development projects in any phase of clinical development, I do think this product will give you previously unavailable insights at speed and at a very competitive cost. We are really focused on broad mechanistic biological coverage with this product. So, of course, we can do, 5,400 with our broad discovery, but a lot of times, you have more focused questions in the neurodegeneration space, but we took great care to cover all of the nodes that are important within this area so that you can simultaneously measure things that you need to measure, like p-tau217, A-beta 4042, NFL, and do some, light version of discovery within this curated list. So I think it's going to meet a really nice niche, be a valuable tool in your tool belt. I think the absolute quantification is really critical if you're looking to compare this data with other data you have that has been delivered in absolute quant. Obviously important to, contextualize this data with other clinical data points you may have already collected, and thinking of cutoffs as they emerge. It's really nice to be able to get that right off the bat. And of course, if you're going to be combining data sets, it eliminates the need for more complex bridging strategies since everything's quantified to a standard. So I think that's really going to be quite nice for everybody. And just to dig a little bit more into the thought process around which markers, we're very fortunate to have a very broad network of KOLs with whom we have strong relationships that informed the panel content. But we also have the data generated from the UK Biobank Pharma Proteomics Project, which has helped guide us towards really proven proteins of interest in this space. So we're excited for you to get your hands on it and see what you can discover and confirm within your own research. So thanks again for your time. I'm going to hand it over to my colleague, Ann Lee, who's the product manager for this Target 48 neurodegeneration panel, and she'll delve into a bit more detail.
Thank you, Evan. I'm so pleased to be here with you all today to share some more about the exciting tool that we've been working on. As I think we can agree, these are exciting times in neurodegeneration biomarker research. One could even say we're at a turning point. After decades of hard work, we now have the tools that can keep pace with the complexity of neurodegenerative disease research. Today, we are introducing a new tool for your tool belt, one that unlocks more data, more clarity, and more opportunity to accelerate biomarker insights in neurodegenerative disease research. For those of you in the audience who may not be familiar with Olink, let me take a few seconds to introduce who we are. Olink is originally a spin-out from an academic lab in Uppsala University in Sweden. And in recent times, we've seen a rapid adoption of the technology. And now we support researchers all over the world. We are an established leader in proteomics and have become part of Thermo Fisher Scientific. And we are truly humbled by the feedback we received from our users and so proud to be serving over 1,000 academic research institutions, biopharma, including the largest pharma companies. And since the first publication of our technology, there are now over 3,000 peer-reviewed articles, which we see as a strong scientific validation of the quality of the solutions that we provide. We've helped analyze more than 4 million samples at this point, and there are many more projects ongoing. At the heart of Olink's contribution to the research community is this innovative proximity extension assay, or PEA for short. PEA enables the simultaneous detection of tens to thousands of proteins in various biofluids. And each PEA assay has a 3-step authentication built in. So 2 matched antibodies must bind to the correct target protein. Each one is conjugated to a complementary DNA oligonucleotide, and these DNA oligotags, as we call them, match together in close proximity, forming a double-stranded DNA molecule for PCR amplification and detection using quantitative PCR. And this rather simple and elegant PEA technology solves many challenges of traditional protein multiplexing: specificity, sensitivity, precision, dynamic range, and throughput, which you will see more about in this webinar later on. The platform requirement is minimal, so you only need 1 microliter of sample for the panel, and you can use a wide variety of sample types, including plasma, serum, dried blood spots, CSF, and dozens of other sample types on this PEA technology. And the low volume requirement and matrix flexibility makes this technology especially great for studies where sample availability is limited or precious, such as many of the neurodegenerative disease studies and clinical trials that are ongoing today. And lastly, but importantly, this technology allows protein quantitative measurements, so readouts in picogram per ml. And with that background, now let's turn our attention to the topic of today's webinar. As we all know, neurodegenerative diseases are some of the most urgent scientific challenges that we face. Already more than 3.4 billion people are directly affected by neurological diseases, and that includes 50 million people worldwide living with dementia, and this number is expected to double every 20 years. But finding preventative strategies and treatments for neurodegenerative diseases is also some of the most complex problems that we face. In recent years, we have seen some truly incredible success. We have seen treatment approvals, but it is clear that our current solutions will not fully resolve the problems that patients and families suffering from these diseases are facing today. Clearly, much more is underway, and the community represented by many people listening into this webinar today is working each in our own capacity to deliver solutions. And it's clear that one critical component to moving faster towards solutions is biomarkers. So those are informative, robust biomarkers that really mirror disease pathology, progression, and supports our development of new treatments. And one of the most useful biomarker types in this context are ones that we can study in biofluid samples. The FDA biomarker approvals in recent years are a true testament to the progress that has taken place in neurology biomarker research. This plot only stretches to 2022 and includes CSF and blood-based biomarkers that are approved by the FDA, and the speed since 2022 is even higher. Interestingly, if we take these selected biomarkers that you saw in the previous slide, those that are involved specifically for dementia work, and we map them onto visualization of the human proteome, for example, here done in Olink Insight. This is what the established fingerprint of dementia looks like. And if we add recent published data on the PEA platform, this is the picture we get. This is the emerging fingerprint of dementia. So all of these dots represent potential protein biomarker candidates. This map is built by funders, patients, researchers, including you and your colleagues, everyone researching and publishing new information. But anyone who has published a study with proteins represented in this graph knows the immense task that remains to take something from an important research finding to an emerging biomarker with early evidence to a biomarker that is useful in the real world. And the focus of many researchers across the world is to move these emerging biomarkers further along that path to clinical relevance and utility. In dialogue with you and listening closely to the situation you're working in right now, you've been telling us that you could move these early biomarker candidates along the continuum toward clinical relevance faster, but your work is currently constrained by a few factors that we've identified in conversation with you and your colleagues. So you have highlighted no robust tool for absolute quantification in plasma, the need to run multiple panels to get to results, and insufficient biomarker breadth. And here is a quote from one of the interviews we conducted in preparing for this. There's a critical need for a tool that enables the detection of key neurodegenerative disease biomarkers for plasma samples to enhance analysis of patient material within clinical trials. A high-performance absolute quantification solution is urgently required to ensure precise, reliable, and reproducible results across and within longitudinal studies. Here. As most of you know, Olink isn't new to neurology. Our technology is trusted and preferred by some of the world's most important neurostudies and consortia. In this graph, we represent them by the 430 plus publications. Those include publications from the UK Biobank proteogenomic work that's really been revolutionizing this space in the last couple of years, as well as publications from the dedicated Coral Consortium focused specifically on neurological conditions and working on the Olink proteomics platform, as well as the fantastic progress made by Octave Biosciences when they analytically validated, clinically validated, and finally launched their multiple sclerosis disease activity test, now in use in clinics across the US. So Olink has provided tools to enable users to pave the way from discovery to clinical translation. And with this longstanding dedication to neurology research exemplified, you won't be surprised that we are well positioned and also committed to trying to help with this request as well. So we've been listening and working hard in the lab. And today we are so proud to present you with the results. Olink Target 48 Neurodegeneration is our newest solution. It can provide you with clarity and complexity in absolute quantification with key markers for profound insights. Now let's look a bit deeper at the tool itself. Target 48 Neurodegeneration gives you simultaneous measurement of 41 key and emerging proteins for neurodegeneration research, requiring only 40 samples per kit and using only 1 microliter of sample. You get your data in picogram per ml readout, and the panel can be run either on your own signature Q100 instrument or at a service provider near you. What is really unique about this new tool? Well, the unique value sits at the intersection of these 3 pillars depicted here in the slide. It's in its ability to simultaneously and confidently measure both key and emerging markers of neurodegeneration with absolute quantification. So now let's explore how this new tool can provide you with previously inaccessible insights. With the Olink Target 48 Neurodegeneration panel, you can achieve precise absolute quantification of these 41 key and emerging biomarkers from microliters of plasma or CSF. And you can compare this data across cohorts, time points, various study centers, disease subtypes, labs, and projects. So that supports a stronger global collaboration and faster progress, and ultimately speeds up our path to clinical translation. What makes this panel truly powerful is that all these 41 key and emerging biomarkers are available together, including the established markers you will appreciate and easily recognize, like NFL, GFAP, p-tau217, A-beta 40, and 42. And this means that you no longer need to piece together multiple panels just to cover the essentials. Instead, everything you need is built to the single scalable assay, and it allows you to track critical biomarkers efficiently and confidently all in one go. This saves your time, cost, and the precious patient samples. And it's not just about efficiency. The panel also delivers mechanistic breadth. This is something we paid special attention to during the development process. So we wanted to give you the ability to dive deeper into the biology that drives neurodegenerative disease. So it spans synaptic proteins, neuroinflammation, clearance pathways, and more, capturing the processes that are most critical to the diseases. And these markers are essential, of course, for studying conditions such as Alzheimer's disease and other dementias, multiple sclerosis, Parkinson's disease, but also many other neurodegenerative and neurological conditions. And some of these proteins and acronyms you will not recognize. So let's look at some higher resolution at the mechanisms in which they are involved. So here's a closer look at those emerging markers that and which disease mechanisms they cover. You see synaptic function and dysfunction, neuroinflammation and immune activation that we touched on before, myelin integrity and axon glial interactions, vascular and neurovascular pathology, as well as proteins involved in protein aggregation and tau and amyloid pathology, oxidative stress and mitochondrial health, and neuroprotection and regeneration. So how did we actually curate the selection of these specific emerging biomarkers? It might be less obvious than how we selected the established ones. Here's an overview of our content strategy, which started with user biomarker requests, specialist interviews, publication review, and database analysis. And then that first wish list was filtered through performance criteria and relevance in plasma, and then further through technical performance assessment that my colleague from R&D, Jenny Rubin, will cover in the next part of the webinar. Here you see the emerging markers broken down by the main source of inclusion rationale that we covered in the previous slide. And you see that we gave substantial weight to biomarkers identified in the large-scale plasma-based neurodegenerative research findings from the UK Biobank. Of course, the thought behind that is to make things more likely to give you a successful result in the end. Now, let's give you a concrete example. So let's zoom in on one of the panel proteins, synaptotagmin 1, or SIT1 for short. This is a presynaptic calcium sensor detectable in human CSF and plasma, and it is one of the very few synaptic proteins found in the UK Biobank to be upregulated in plasma 15 years prior to incident AD diagnosis. And it also has been shown to have a continued increase during disease course. And in the publication highlighted in this picture by Zhang et al., they leveraged the UK Biobank Olink data and identified SIT1 as a causal protein for AD. And this gave us the confidence that this is a good protein to include because it does have a the potential to reflect early synaptic dysfunction, something that's very critical for the field and that many of us are trying to enable identification of, and then also tracking and therapeutic monitoring. And here a quote from the paper saying that the earliest and sustained alterations throughout the course of AD among thousands of proteins we studied beside GFAP and NFL, as you can see here in the plot. So for whom did we develop this tool? Well, for those working in academia, for clinical researchers, and those working in drug development programs in pharma companies, and for diagnostics developers. And now let's further detail some examples of how researchers have indicated that they want to apply this tool in their work. So firstly, if you are working in the academic setting, the way that you can use this tool is, for example, to use the mechanistic breadth and absolute quantification to do biomarker candidate comparisons across time in your studies or across cohorts and diverse populations. This is critical for understanding the early pathology that you want to study and advancing the biomarker candidates in a global context. And it also supports the validation across your collaborator network. If you are based in more of a clinical setting in a university hospital, for example, this research tool enables you to study heterogeneous disease progression and treatment response in your study participants by measuring key and emerging markers in picogram per ml readout, which is intuitive to interpret and all in one run. It also spares the precious samples and funding while confidently tracking biomarker candidate levels with patient trajectories that you might be already studying. If you are instead working in a biopharma context, this 14-protein panel can really represent the sweet spot to efficiently validate exploratory biomarker hypotheses, which speeds up the time-sensitive transition from early to late phases in drug and diagnostic co-development programs. And by running all of these markers in one panel, that can save you time and cost and patient samples, getting you faster to well-informed decisions within the drug program. If you detail this further, depending on your role in that work, you can think of it from a lab context that you would want to check if your biomarker candidate correlates with drug activity. Does it correlate or inform dose response? Does it track well with disease progression or identify the right patients? And this tool can then provide you the power that you need to figure all of this out in a single run while also saving you resources in your day-to-day. And if you are instead working in a CRO context, this is something that you can offer to your pharma customers to, for example, pool their phase 2 clinical trial samples for retrospective analysis in the panel. And then that data will help your pharma customers de-risk their biomarker selection for their phase 3 trials faster. And that's a great way to support your pharma partners and to accelerate their drug and DX development. So these are some examples. I'm sure this is not an exhaustive list, but this is what we've heard from you, and this is how we imagine that you will use the product. Now to tie things together, I'd like to summarize it with this slide. The panel combines the ability to simultaneously and confidently measure both key and emerging biomarkers for neurodegeneration with absolute quantification, providing a unique and innovative tool for you to find previously inaccessible insights and really translatable insights in neurodegeneration research. And should you want to delve deeper into other aspects that are also important for neurodegeneration research, for example, inflammation, you can add a cytokine panel that is also available in Apps Quant. Or if you want more custom markers, we have an offering where you can add or you can combine this target neurodegeneration panel with a flex offering, as well as if you want to build a dedicated panel, then we have a focus offering that's not depicted in the graph, but we can, of course, help you with those discussions as well. Now, I'd like to sum things up and hand over to my colleague, Jenny Rubin, the R&D scientist in charge of the project and who is keen to share some performance data with you. So Jenny, over to you.
Thank you, Anneli, for illustrating the value of the new Target 48 neurodegeneration panel. From my side, I am delighted to now share some of the performance data generated in the development of the panel. So firstly, I would like to share some details on the verification process. Assay development at Olink follows a 15-factor verification process, as shown in this slide. Each assay is assessed for its sensitivity, and this is done by analyzing recombinant antigen curves. Specificity, we also analyze recombinant antigens to ensure there is no cross-reactivity. For detectability, we analyze several different matrices, such as plasma and CSF and serum. For each assay, the dynamic range is established and also the optimal sample dilution. But today, I will share some data from the final verification of the Target 48 neurodegeneration panel. So Target 48 neurodegeneration is an absolute quantification panel. So it means that the output is picogram per ml, and MPX data is also provided. All Target 48s are run in a 48 times 48 format, meaning 48 samples are assayed, of which 8 are external controls, and up to 48 assays are included in the panel. So the external controls consist of calibrator that is used for data normalization, the sample control, that's a QC sample included in each run. And these two components are both tailor-made to have a signal in the middle of the range for all assays. The negative control is used for load calculations. To calculate picogram per ml values, MPX data is converted to picogram per ml using an antigen standard curve that is generated during product verification, and I will show an example of this soon. So absolute quantification at Olink means data conversion using an antigen standard curve. So this is an example of an antigen standard curve. We run a 24-point calibrator curve. So this is done during panel verification. So this 24-point calibrator curve consists of recombinant antigens. And this standard curve is then the basis for the 4PL fit of each assay and the generation of absolute quantification data. So 4PL parameters are established for each assay, and the lower level of quantification and upper level of quantification is then set based on the shape of the antigen curve. And data is normalized with the calibrator I mentioned previously to adjust for variance between runs, inter-run variability. So Olink Target 48 neurodegeneration is a panel that is optimized for use with plasma samples, but to estimate detectability, several different sample matrices were analyzed. The verification cohorts include matched EDTA plasma and serum from 15 controls, EDTA plasma samples from 65 patients who have been diagnosed with various neurological impairments, such as Parkinson, ALS, Alzheimer's, MS, and traumatic brain injury, and also a cohort of CSF samples, both from control donors as well as CSF from patients with these neurological impairments have been analyzed. So in this graph, we illustrate the dynamic range of each assay. So the dynamic range, by that we mean the quantifiable range of each assay in the panel. So in these graphs, the light blue bar shows the assay range, the range between LLOQ, which is the lowest level of quantification, and to the highest level of quantification, that's the ULOQ. In dark blue, we show the distribution of the analyzed samples. So the left graph illustrates the EDTA plasma samples of controls. But since there's a lot of interest to also analyze this panel for CSF samples, we also have the right graph illustrating all CSF samples analyzed in the verification. So this is the range of both the control samples as well as the pathological samples. And this data will be available in the webpage in the data validation package. So it's easy to zoom in there and look and investigate assay by assay what detectability we had in our verification. Precision is something we think is quite important in our panels. So we have our sample control. This is a reference sample that is analyzed in triplicate in every PEA run. So this sample is designed to have a signal in the quantifiable range of each assay, and it is then used for QC of each assay in each run. So in this graph, the mean intra and inter CV per assay is presented, and data is calculated on sample control data from the verification runs. So a total of 24 runs performed by 3 different operators. We had excellent performance on sample control. So the intraplate CV was below 10% for all assays, and the interplate CV was below 15% for all assays. Now I want to show you some data on specific assays. So here we have assay NEFL, which is a new assay for the absolute quantification library in Olink. To the right is the 24-point calibrator curve illustrating the range of the assay. And we had excellent performance when we measured precision of the sample control. Both the mean intra-CV as well as the inter-CV was 6% in the verification. So these CV numbers are from the sample control run in the verification. GFAP is another assay that is new to our absolute quantification library in Olink. Again, we have the 24-point calibrator curve illustrating the range of the assay. Really good performance on sample control with intra-CV at 6%, mean overall runs, and the mean inter-CV at 7%. VBOX, a new emerging marker involved in protein aggregation, and it's been linked to both Alzheimer's and Parkinson's disease. Again, we have the 24-point calibrator curve illustrating the range of the assay, but to the right, we also have a sample distribution plot. So this illustrates the measured picogram per ml values of all the EDTA plasmas in the plasma samples in the verification. So to the left in the graph in light blue, we have the control samples, and then the other groups are different neurological diseases. So while this is a small sample set, we can still see an increase in signal in the neurological diseases. And we also have p-tau217. Again, an assay that is new to the absolute quantification portfolio of Olink. Phosphorylated tau217, which is a key blood biomarker for diagnosis in Alzheimer's disease. You can see the 24-point calibrator curve illustrating the range of the assay. We have a really nice range from 0.19 picogram per ml up to close to 5,000 picograms per ml as the top limit of quantification. Really good performance on sample control with the mean intra-CV of 6% and the same number for the inter-CVs. Again, we have a sample distribution plot to the very right on the slide, where we have control samples in light blue to the left and different neurological diseases. I just want to highlight that here we actually have a linear scale on the y-axis to illustrate the levels of protein in the different samples. So we can see a difference in signal from normal donors to the pathological samples in plasma. Olink's p-tau 217 assay was also compared to other providers. So the Olink P assay performance was verified by comparing data to two well-known providers. The graphs to the right illustrate Olink PA data on the x-axis correlated to company A on the y-axis in the top graph and company B on the y-axis in the bottom graph. So a mix of both EDTA plasma and CF samples were analyzed, in total 30 samples. And it was a mix of samples from control donors as well as pathological samples. The runs were performed by external service providers. And we are quite happy to illustrate that Olink's PA assay correlates really well to both these methods. So with that, I hope that I've been able to represent some critical parameters and performance metrics of our analytical verification process. The full details on the verification data is available in our data validation package on olink.com. And with that, I would like to hand it back to Evan for his closing remarks.
Thanks again for your time. I hope it was time well spent and that you're as excited as we are about the opportunities that are ahead of us with this new Target 48 neurodegeneration panel. Again, I think it's a great thing for our company, but it's a greater thing for the neurodegeneration research community. There's a lot of products in the space, but I think compromises have been the norm. We're really hoping to eliminate that and give you a super versatile, very powerful tool. We know it's been challenging, right, to push from this discovery to diagnostic phase. The translatability, having to use different platforms creates real challenges. And, we're thrilled to be your partner on any part of the journey that you're on. And really our mission, right, consistent with Thermo Fisher's mission is, to make the world healthier, cleaner, and safer. I do believe that, the neurodegeneration field, there's a massive need in the field as, the aging population is going to need better treatments, better biomarkers. It's great that treatments are emerging. And our passion is to provide tools that will help not only provide evidence for next generation therapies, but hopefully provide some biomarkers for early detection, disease progression, and perhaps some efficacy measurements as new therapies come to market to help so many people that are going to benefit from these advances. So we're hoping to bring clarity to complexity. Neurodegeneration is a super complex process, very, very hard to access, tissue, obviously. So we're hoping that we're part of the tool set that can do peripheral measurements that are incredibly impactful as you learn more about your diseases of interest and try to advance better therapies, better tests, and better outcomes for patients ultimately. So that's all I've got for today. Just as a reminder, this panel is available right now, and you can order it and get delivery by the end of the month. We have a global set of service providers that will be ready to help you with this product as well. They'll have it on stock if you need it. So that would give you easy access either through your own lab or through a wide network of providers. You can submit your request on www.olink.com. I do want to recognize many of the team members here at Olink that have worked really hard for a long time to build this product. I want to thank our KOL partners and customers that have helped us shape this product. I think that's always going to lead to a better outcome. This was not built in isolation. This was built with the guidance from people that know a lot more about this than us. So I think that is fantastic. And thanks for inspiring us to build better tools to enable the next generation of discoveries. Thanks again, and looking forward to seeing all the wonderful work that you can do with this product. Thanks.
All right. Thank you all very much for your very informative presentations. We will now go ahead and move into the live Q&A portion of the webinar. So if you happen to have a question that you would like to ask, please go ahead and do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. Also, I quickly want to mention that Olink has an upcoming online conference, Olink Proteomics World, October 8th, 2025. During that meeting, Annalee will interview David Walk from Penn Neuroscience Center regarding the evolving role of biofluid biomarkers in neurodegenerative disease reach. So please feel free to register for that meeting as follow-up on this session. Okay, let's go ahead and dive into the Q&A here. We are going to go ahead and start off with this question for Annalee. And Annalee, this one says, why is the number of people affected by dementia expected to increase so much in the future? Has any clear cause been identified?
Well, thank you, Sydney, and thanks for your question. This is an important one. So the rise in numbers for dementia is largely due to the aging population, essentially that we live longer. So age is the main risk factor for dementia and neurodegenerative diseases. So this is the main driver of this increase in numbers.
Okay, wonderful. It looks like this next question here is for Jenny, and this one says, maybe a more technical question, but has it been tested whether changes in pH given in a sample affect the binding affinity of the antibodies to the individual antigens prior to qPCR?
Right. So for this panel in particular, we run plasma neat. So we don't expect any impact basically on pH. We kind of expect it to be the same in all samples. However, we do use recombinant antigens in our standard curves, and different antigens or proteins are obviously affected by pH. It could affect the folding, etc. So for individual recombinant antigens, yes, I do agree that pH could be a factor. In our testing, we do multiplex of the recombinant antigens, and we keep them in the same environment with the same pH basically all the time. Yeah.
Perfect. Okay, it looks like we have another two-part question here for you, Jenny. And this one says, any experience with testing interstitial fluid? Which sample protein concentration is required? Is it the same for all sample types?
Right. So we haven't tested that for this particular panel development, but I know it has been done before in previous development projects. So I think there is data and help to get from the support team. So that would be my suggestion, to reach out to the Olink support team to get help with protocols and recommendations.
Thanks, Jenny. I can also add a little bit to that. So there are researchers that have run EVs using our other products in the past. And on our website, you can find publications link, and you can filter through. And we're also happy to help you connect you to those resources. So we think EVs are a very interesting field, and I hope more people will explore it further. There are a lot of biomarkers that are believed to be contained within those vesicles. So look forward to seeing the results of that work.
Okay, fantastic. Next question we have here for Annalee. It says, what is the test duration?
Thank you for this question. So it's 2 hours hands-on time and data within 24 hours.
Perfect. Also for Annalie, a two-part question here. This one says, can you provide a list of service providers and where can I send my samples for analysis?
Thank you. That's obviously a very important question for next steps. So the best way to go about this, I think, is to go on our website and search the service providers that you can there filter by region and country so that you can find the one closest to you. We can also, of course, provide additional guidance if you need. So I hope that gives you the answers.
Okay, wonderful. Annalie, this one is also for you. It says, do we have plans to publish case studies or white papers demonstrating real world applications and findings from using this panel?
Thank you. That's also a great question. So the data that Jenny showed was obviously from our internal verification studies, and we always have white papers that we publish together with our products, and that's the intention here as well. So yes.
Okay, wonderful. Let's see here. Jenny, it looks like this one is for you. It says, has the neurodegeneration panel been tested on CSF?
Yes, so in our internal verification, we have analyzed both CSF and plasma. So this is a plasma panel, so all technical performance has been focused on plasma samples, but for detectability, we have analyzed a number of CSF samples also. Some data is available already in the PDF available on the website, and in the upcoming assay pages, there will be more information also on detectability in CSF.
I can jump in and add one more little piece there. Thank you, Jenny. That's the perfect answer. And I just wanted to say that obviously a lot of the biomarkers are working, are moving from CSF into blood, which has been the case for some of the recent advancements. So we have gotten a lot of feedback on interest from other people asking similar questions. So we will be as transparent as we are and just share everything that we've done in-house so that you can see how it performs in our hands. That said, you know, just like Jenny said, the panel is optimized for plasma, so that's where we've carried out the most tests and where we have most information. But for CSF, we have a lot of experience with all our other products, and we've always seen that the performance is really good. So it's just that the ranges of the proteins are different, so you expect to see different levels in those two matrices.
Okay, wonderful. It looks like this next one that we have here is for Jenny. It says, thank you for an informative webinar. It says, how is your method of absolute quantification different from your verification process in the relative MPX quantification panels? The method looks similar.
It is very similar, so it makes sense that it looks similar. So basically what makes the absolute quantification a bit different then is in the verification phase, we run these 24-point standard curves of recombinant antigens. We have a calibrator, which is a tailor-made sample designed to have a signal in the range of all assays. And this is used for the normalization of data. So in our absolute quantification, we do this conversion from MPX to picograms per ml. So yes, a lot of the tests and the processing we do is very similar, but in the absolute quantification panels, we have our calibrator to enable our absolute quantification.
Perfect. Okay, it looks like this one also is for you, Jenny. It says, great panel. How are you going to keep the standards fully absolute quantitative across different lots as they are based on a standard curve with recombinant material? How does your standard compensate for matrix effects?
A lot of questions in one. So basically what we do is we have, we put a lot of effort into making sure we have lot-to-lot consistency. So as we just talked about. We have our calibrator for inter-run variability, but then we also do check for lot-to-lot variability, basically running 2 lots of the product at the same time. We analyze the antigen standard curve. We analyze biological samples to make sure that we align the output as much as possible. So a bridging factor is calculated in that step, and that is applied on all the data going forward to make sure that regardless of lot, you will have the same output in your samples. Was there another part in the question there, or did that cover it?
Let me take a look here. I think you covered all of it.
Thanks.
Perfect. Okay, so we have 2 more questions here. We will go ahead and wrap up with these final 2. This next one is for Annalie, and it says, is there a software available to deal with results as for Explore Panel?
Yes, there's a software available that comes with the product. So that's a very important question. Thank you for asking.
Okay, wonderful. And we will go ahead and wrap up today with this final question. This one also is for Annalie. It says, do you try to cover the broadcast biological pathway? Or I'm sorry, do you try to cover the broadest biological pathway or to strengthen specific pathway that are already well known?
Thank you. That's also an excellent question. So the way we try to work with this in development of this new tool is to really give you both, essentially. We wanted you to have the key biomarkers that are where there's a breadth and wealth and depth of literature, and you know how you expect them to perform in your samples based on the disease pathology or the treatment that you're that the samples that the patients have been exposed to from whom you have samples. And the other aspect that we really wanted to cover was this huge group of emerging markers where synaptic proteins are very important for the field moving forward, and also a lot of other pathways that we've highlighted in the previous presentation. So we really wanted to strike that balance between having things where you know what to expect, so those critical sort of big 5 biomarkers, if you will, and to combine those with all of these emerging ones where you can then interpret your findings in a more reliable way, because you can directly relate them to those other values. So we wanted to strike that balance between what you already know and the exciting unknowns where you really want to uncover more information about those pathways. I hope this answered the question.
Perfect. Thank you so much. Thank you both again for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, Olink Proteomics, for underwriting today's educational webinar. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. As a quick reminder, this webinar can be viewed on demand. We will go ahead and alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. All right, until next time, thanks so much, everybody. Goodbye.
Thank you. Bye.